Alright, welcome everybody. This is going to be Chapter 2, an introduction to genes and genomes. I'm going to run through all the vocabulary for this chapter. I'm just going to read off the names and the definition. That's all this video is going to be. I'm not going to read the entire chapter. There's already a video that reads the entire text of this chapter. So we're going to just jump right into it. We can just follow along on this screen. Okay. The first term that we're going to cover is they're going to be in blue. Okay. In section 2.1 we have deoxyribonucleic acid, or DNA, and this is double-stranded nucleic acid consisting of bases, adenines, guanines, cytosines, thymines. Also, in addition to the bases, sugars, which is deoxyribose, and phosphate groups arranged in a helical molecule, inherited genetic material that contains genes, which direct the production of proteins in a cell. So again, DNA is double-stranded nucleic acids consisting of bases, sugars, and phosphate groups arranged in a helical model. Okay. The next term will be plasma membrane, which is a double-layered structure consisting of lipids, proteins, and carbohydrates that defines the boundaries of a cell, performs important roles in cell shape, and regulating transport of molecules in and out of a cell. Next is the cytoplasm. The cytoplasm is the inner contents of a cell consisting of fluid cytosol and organelles. Its boundaries are defined by the plasma membrane. Alright, next is the cytoplasm. The cytoplasm is the inner contents of a cell consisting of fluid cytosol and organelles. Its boundaries are defined by the plasma membrane. And here we have organelles. Organelles are small structures in the cytoplasm of eukaryotic cells that perform specific functions. Prokaryotic cells. Prokaryotic cells are cells that lack a nucleus and membrane-enclosed organelles. The only examples are bacteria and archaea. And they mean before the nucleus. That's important. Nucleus. Nucleus is membrane-enclosed organelle that contains the DNA of a eukaryotic cell. Domain archaea. Domain archaea is a classification or domain of prokaryotic cells. A common feature of these microbes is that they live in extreme environments, for example, boiling hot springs. Next we move to the eukaryotic cells. Definition here. Cells that contain a nucleus, including plant and animal cells, such as protists and fungi. Or not such as, but including plant and animal cells, protists, and fungi. Those are eukaryotic cells. Here's a picture of an example. Next we're going to do adenosine triphosphate, ATP, a nucleoside triphosphate that contains the nitrogenous base adenine. ATP is the primary form of energy used by living cells. Next is the cytosol. Cytosol is gel-like, nutrient-rich, aqueous, or water-based fluid of the cytoplasm. Alright, we want to know the structure and function of eukaryotic cells. Yeah, I'll go ahead and read these. The part, structure, and function. Plasma membrane made of a double layer of lipids, primarily phospholipids and cholesterol, within which proteins are embedded. Proteins may extend entirely through the lipid bilayer or protrude on only one face. Externally facing proteins and some lipids have attached sugar groups. The functions of the plasma membrane serves as an external cell barrier, acts in the transport of substances into or out of the cell, maintains a resting potential that is essential for the functioning of excitable cells. Externally facing proteins act as receptors for hormones, neurotransmitters, and so on, and in cell-to-cell recognition. Cytoplasm. We already got to that, but cellular region between the nuclear and plasma membranes consists of fluid cytosol containing dissolved solutes, inclusions, which are stored nutrients, secretory products, pigment granules, and organelles, the metabolic machinery of the cytoplasm. Cytoplasmic organelles. The mitochondria. Rod-like double membrane structures. The inner membrane is foldable into projections called cristae, and the function is the site of adenosine triphosphate synthesis, and is also known as the powerhouse of the cell. That's the mitochondria. Okay, ribosomes. Structure. Dense particles consisting of two subunits, each composed of ribosomal RNA and protein. They're free or attached to the rough endoplasmic reticulum, and their function is the site of protein synthesis. The rough ER, or rough endoplasmic reticulum, is the membrane system enclosed in a cavity, the cisterna, and coiling through the cytoplasm, externally studded with ribosomes. Sugar groups are attached to proteins within the cisternae. Proteins are bound in vesicles for transport to the Golgi apparatus and other sites. External face synthesizes phospholipids and cholesterols. Smooth ER is a membranous system of sacs and tubules free of ribosomes, the site of lipid and steroid synthesis, lipid metabolism, and drug detoxification. Golgi apparatus, a stack of smooth membrane sacs and associated vesicles close to the nucleus. Packages, modifies, and segregates protein for secretion from the cell, inclusion in lysosomes, and incorporation into the plasma membrane. Lysosomes. Membranous sacs containing hydrolases, digestive enzymes, and it serves as the site of intracellular digestion. Peroxisomes, membranous sacs of oxidase enzymes, and the function is the enzymes detoxify a number of toxic substances. The most important enzyme, catalase, breaks down hydrogen peroxide. Microtubules. Cylindrical structures made of tubulin proteins. They support the cell and give it shape, involved in intracellular and cellular movements. They form the centrioles. Microfilaments are fine filaments of the contractile protein actin. They are involved in muscle contraction and other types of intracellular movement. They help form the cell's cytoskeleton. Intermediate filaments. Protein fibers. Composition varies. The stable cytoskeleton elements resist mechanical forces acting on the cell. Centrioles. Paired cylindrical bodies, each composed of 9 triplets of microtubules. They organize a microtubule network during mitosis to form the spindle and asters They form the bases of cilia and flagella. The cilia. Short cell surface projections. Each cilium is composed of 9 pairs of microtubules surrounding a central pair. They move in unison, creating a unidirectional current that propels substances across cell surfaces. Flagella. Like cilia but longer, the only example in humans is the sperm tail. They propel the cell. And the nucleus, which is the largest organelle, surrounded by the nuclear envelope, contains fluid nucleoplasm, nucleoli, and chromatin. The function is the control center of the cell, responsible for transmitting genetic information and providing the instructions for protein synthesis. Next up is the nuclear envelope, double membrane structure pierced by the pores. They are the outer membrane continuous with the cytoplasmic endoplasmic reticulum. Separates the nucleoplasm from the cytoplasm and regulates the passage of substances to and from the nucleus. Nucleoli. The dense, spherical, non-membrane bound bodies composed of ribosomal RNA and proteins. They serve as a site of ribosome subunit manufacture. Chromatin. Granular thread-like material composed of DNA and histone proteins. They serve as their function is DNA contains genes. Central vacuole, only in plant cells, is a large membrane enclosed compartment used to store ions, waste products, pigments, and protective compounds. Chloroplasts, plant cells only, membrane enclosed organelle containing stacks or stacked structures, grana, of chlorophyll containing membrane sacs called thylakoids, surrounded by an inner fluid called the stroma. The function of chloroplasts is the site of photosynthesis. Just previously we mentioned the nuclear envelope. A double-layered membrane is typically the largest structure in an animal cell. Okay, next up, from section 2.2, the molecule of life, we have proteases, protein digesting enzymes. Next, ribonucleic acid, RNA, single strands of nucleotides produced from DNA. Different types of RNA have important functions in protein synthesis. We'll learn about the different types of RNA shortly. Nucleic acids. Molecules composed of nucleotide building blocks. Two major types are DNA and RNA. Next we get into section 2.3. And our first term here is transformation. The process by which bacteria take in DNA from the surroundings. Term also used to define changes that cause a normal cell to become a cancer cell. Next is DNase. DNA degrading enzyme. Pentose sugar. A 5-carbon sugar, important components of DNA and RNA nucleotide structure. The pentose sugar deoxyribose is contained in DNA nucleotides. The pentose sugar ribose is contained in RNA nucleotides. So, 2 pentose sugars are mentioned. Deoxyribose for DNA and ribose for RNA. Important. Next is nitrogenous base. Important component of DNA and RNA nucleotides, often simply called a base. Nitrogen-containing structures include double-ring purines, which include the bases adenine and guanine, and single-ring pyrimidines, which include the bases cytosine, thymine, and uracil. Okay, and here's adenine. Adenine, abbreviated capital A, is a purine base present in DNA and RNA nucleotides. Cytosine. Cytosine, abbreviated C or capital C, is a pyrimidine base present in DNA and RNA nucleotides. I have X-ray crystallography, fast X-ray analysis of protein structure by using algorithms and data analysis. Next will be helix. Helix is one of two secondary structures of proteins. Next is phosphodiester bonds. Phosphodiester bonds are covalent bonds between the sugars of one nucleotide and the phosphate group of an adjacent nucleotide. They join nucleotides within strands of DNA and RNA. Next is polarity. Polarity refers to the 5' and 3' ends of DNA and RNA molecules. Complementary base pairs refer to the nucleotides adenine A and thymine T, or uracil U, when referring to RNA, and guanine G and cytosine C in a DNA molecule. A and T and G and C nucleotides join by hydrogen bonds to complement each other. I missed 5' end. I'm going to backtrack. 5' end, the end of a strand of DNA or RNA in which the last nucleotide is not attached to another nucleotide by a phosphodiester bond involving the 5' carbon of the pentose sugar. 3' end, the end of a strand of DNA or RNA in which the last nucleotide is not attached to another nucleotide by a phosphodiester bond involving the 3' carbon of the pentose sugar. Antiparallel or antiparallel refers to the 5' and 3' directionality of strands of DNA in which the two strands joined together by hydrogen bonds between complementary base pairs are oriented in opposing directions with respect to their polarity. Next is gene. A gene is a specific sequence of DNA nucleotides that serves as a unit of inheritance. Genes govern visible and invisible characteristics, which the invisible characteristics are traits of living organisms in large part by directing the synthesis of proteins in a cell. And next we define traits. Inherited features of an organism such as skin color and body shape. Next is chromosomes. highly folded arrangements of DNA and proteins. They pack DNA to allow even separation of genetic material during cell division, and that again is chromosomes. DNA binding. DNA binding domains. Regions with folded structural arrangements of amino acids called motifs that interact directly with DNA. Histones. Histones are DNA binding proteins that are important for chromosomal structure. Chromatin. Chromatin. Strings of DNA wrapped around proteins present in the nucleus of eukaryotes and the cytoplasm of prokaryotes. Coils tightly together to form chromosomes during cell division. Maternal chromosome. Maternal chromosomes, the 23 chromosomes inherited from your mother. Paternal chromosomes. Copies of chromosomes inherited from the father. Homologs. Actually, no. Homologous pairs. Pairs of chromosomes or genes that occur in organisms with multiple sets of chromosomes. Homologs. Related genes in different species. Related genes in different species. Genes share sequences similarity because of a common evolutionary origin. Autosomes. Chromosomes whose genes are not primarily involved in determining an organism's sex. Chromosomes 1 through 22 in humans are known as autosomes. Sex chromosomes. They contain the majority of genes that determine an organism's sex. The X female and Y male chromosomes in humans. So again, the X sex chromosome is for female, and the Y sex chromosome codes for males. Next is gametes. Gametes are haploid cells, often called sex cells. They include human sperm and egg cells, ova. Gametes join together during fertilization to form a zygote. Could also see meiosis. Haploid number, N, denoted by lowercase n. A single set of chromosomes, N, often used to describe cells with a single set of chromosomes. Haploid. Next is somatic cells. Somatic cells are all cell types in multicellular organisms except for gametes, which the gametes are sperm and egg cells. Diploid number, which is denoted by 2n. Two sets of chromosomes, 2n, often used to describe cells with two sets of chromosomes, typically one set from each parent. Next is the P arms or Q arms. Q arm is a long arm of a chromosome, and in contrast, the P arms are the short arms of a chromosome. Telomeres. Telomeres are the end structures of a eukaryotic chromosome. In humans, they consist of specific repeating sequences of DNA, like TT, A, G, G, G. Next is sister chromatids. Sister chromatids are exact copies of double-stranded DNA molecules and proteins, chromatin, joined to form a chromosome. Then we have centromere, constricted region of a chromosome formed by intertwined DNA and proteins that hold two sister chromatids together. We already defined the P arm. Spectral karyotyping, a technique involving probes specific for each chromosome that fluoresces different colors to identify chromosomes according to their color pattern. And next is mitosis. Mitosis is a nuclear division process that occurs during cell division in eukaryotic somatic cells and prokaryotes. Mitosis is divided into four major stages: prophase, metaphase, anaphase, and telophase, or telophase. Mitosis results in the even separation of DNA into dividing cells. And next is meiosis. Meiosis is a nuclear division process that occurs during formation of gametes. Meiosis involves a series of steps that reduce the amount of DNA in newly divided cells by half compared with those in the original cell. Next is zygote. Zygote is a diploid cell formed when haploid gametes, such as sperm and egg cell, unite. Semi-conservative replication, process by which DNA is copied. One original, which is the parent, DNA molecule gives rise to two molecules, each of which has one original strand and one new strand. DNA helicase, or helicase, enzyme that separates two strands of DNA molecule during DNA replication. Single-strand binding proteins, proteins that bind to unravel single strands of parental DNA during DNA replication to prevent DNA from reforming double strands before being copied. Origins of replication. Specific locations in a DNA molecule where DNA replication begins. RNA primers. RNA primer is a short single-strand RNA sequence that binds DNA and is used to start DNA replication by DNA polymerase. Primase. Primase is an enzyme that adds small RNA segments to a single strand of DNA as an early and necessary step for DNA replication. DNA polymerases. DNA polymerases are key enzymes that copy strands of DNA during DNA replication to create new strands of nucleotides. These have important applications for synthesizing DNA in molecular biology experiments. DNA polymerase III. One form of DNA polymerase. The primary DNA polymerase which copies DNA in prokaryotes. Leading strand. The leading strand defined. During DNA replication, the strand of newly synthesized DNA that is copied by DNA polymerase in a continuous fashion, 5' to 3', into the replication fork. Lagging strand. During DNA replication, the strand of newly synthesized DNA that is copied by DNA polymerase in a discontinuous or interrupted fashion, 5' to 3', away from the replication fork, as a series of short DNA pieces called Okazaki fragments. DNA ligase is an enzyme that forms covalent bonds between incomplete or Okazaki fragments of DNA during DNA replication. Routinely used for joining DNA fragments in recombinant DNA experiments. Genomics is the study of genomes. In a genome, we'll just add that in, all of the DNA in an organism's cells is called the genome. Okay? Next, we define messenger RNA. Messenger RNA, also known as mRNA, a template for protein synthesis, is an exact copy of a gene, contains nucleotide sequences copied from DNA that serve as a genetic code for synthesizing a protein, and is then bound and read by ribosomes to produce proteins. Translation. Translation defined is the synthesis of proteins from genetic information in messenger molecules or mRNA molecules. Translation occurs in the cytoplasm of all cells. This is a very important diagram. Okay. RNA polymerase. Copies of RNA from a DNA template. RNA polymerases are different forms of RNA polymerases synthesize different types of RNA. Alright, next is promoter. And this deals with transcription. Promoter. Specific DNA sequences adjacent to a gene that direct transcription, which is RNA synthesis, is the binding site of RNA polymerase to begin transcription. Transcription factor, or factors. DNA binding proteins that bind promoter regions of a gene and stimulate transcription of a gene by RNA polymerase. Enhancers. Enhancers are specific DNA sequences that bind proteins called transcription factors to stimulate, enhance transcription of a gene. Template strand. Template strand. After RNA polymerase binds to a promoter, it unwinds a region of the DNA to separate the two strands. Only one of the strands, called the template strand, which is the opposite strand, is called the coding strand, is copied by RNA polymerase. So again, only one of the strands, called the template strand, is copied by RNA polymerase. The coding strand is not copied by RNA polymerase. Transcription factors. Oh no. Transfer RNA. Transfer RNA are, or tRNA, small RNA molecules that transfer amino acids to a ribosome during protein synthesis. tRNA binds to specific codons in mRNA sequences during translation. Ribosomal RNA, or rRNA. Small RNA molecules that are essential components of ribosomes. Next is microRNAs, or miRNAs. A new class of non-protein coding RNA molecules. MicroRNAs are part of a rapidly growing family of small RNA molecules, about 20 to 25 nucleotides in size, that play novel roles in regulating gene expression. Primary transcript, or pre-mRNA. My dog's snoring. Primary transcript pre-mRNA. Initial mRNA molecule copied from a gene in the nucleus of eukaryotic cells undergoes modifications processing to produce mature mRNA molecules that enter the cytoplasm. Next is RNA splicing. RNA splicing, the removal of non-protein coding sequences, which are introns, removal of non-protein coding sequences, called introns, from primary transcripts, or pre-mRNA, and joining of protein coding sequences, which are called exons. Okay, so introns. Non-protein coding sequences in eukaryotic genes and primarily transcripts that are removed during RNA splicing. Exons. Exons are protein coding sequences in eukaryotic genes and mRNA molecules. Alternative splicing. Splicing sometimes can join certain exons and cut out other exons, essentially treating them as introns. This process creates multiple mRNA of different sizes from the same gene. Each mRNA can then be used to produce different proteins with different, sometimes unique, functions. Alternative splicing allows several different protein products to be produced from the same gene sequence. Next is polyadenylation. Polyadenylation, addition of a short sequence or tail of adenine, A, nucleotides to the 3' end of an mRNA molecule. It occurs during RNA splicing in eukaryotes. Poly A tail is important for mRNA stability in the cytoplasm. Next is ribosomes. Organelles composed of ribosomal ribonucleic acid, or rRNA, and proteins assembled into packages called subunits. Ribosomes bind to mRNA and tRNA molecules and are the site of protein synthesis in prokaryotes and eukaryotes. Genetic code. Genetic code, information contained in bases of DNA and RNA that provide cells with instructions for synthesizing proteins. They consist of 3 nucleotide combinations called codons. Next is amino acids. Amino acids are the building blocks of protein structure. Combinations of 20 different amino acids can join together by covalent bonds in varying order and length to make a polypeptide. Next is polypeptide. A chain of amino acids joined by covalent peptide bonds, usually greater than 50 amino acids in length. Next is codons. Codons, or a codon, comprises a combination of 3 nucleotide sequences in an mRNA molecule. With few exceptions, each codon codes for a single amino acid. 64 possible codons compose the genetic code of mRNA molecules. So there's 64 codons and there's only 20 amino acids. So that means that not all 20 have 3 codons. There's some amino acids that have like 3 or 4 different codons, but there aren't that many of those type. But this table right here is actually really important. You can see these UUU is an example of a codon. UUC is a codon. These 2 different codons code for the same one amino acid. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. But I ended up counting methionine for the amino acid, but it's actually a start codon, so that's really important. AUG, that's a very important one. And then the stop codons, UAA and UAG, those are the stop codons. So this table here, not something that you have to memorize, but can definitely help identify during the sequencing part. But we need to define codons right here. A codon comprises a combination of 3 nucleotide sequences. Oh, actually no, I just defined it. Sorry. Okay, the A, amino acyl site. A, amino acyl site of a ribosome. Portion of a ribosome into which amino acyl tRNA molecules bind during translation. the P peptidyl site. P peptidyl site of a ribosome, portion of a ribosome into which peptidyl tRNA molecules bind during translation. And the E site, a groove through which tRNA molecules leave a ribosome. Okay, amino acyl transfer RNA. Amino acyl tRNA. Transfer RNA, tRNA molecule with an amino acid attached. Okay. Amino acyl transfer RNA. So it just has an amino acid attached. Anticodon. 3 nucleotide sequence at the end of a tRNA molecule. During translation, the anticodon binds to a specific codon in an mRNA molecule by complementary base pairing. Peptidyl transferase. Peptidyl transferase is the RNA enzyme that is part of the large ribosomal subunit, catalyzes the formation of peptide bonds between amino acids during translation. Peptidyl tRNA. The tRNA molecule attached to a growing polypeptide chain, located in the P site of a ribosome during translation. Gene expression. Gene expression is a term generally used to describe the synthesis or expression of RNA by a particular cell or tissue. For instance, if mRNA for the fictitious gene corn is detected in a tissue by PCR or northern blot analysis, that tissue is said to express the corn gene. Next is gene regulation. Gene regulation is a general term used to describe processes by which cells can control gene activity or expression, RNA and protein synthesis. Transcriptional regulation. Transcriptional regulation is a form of gene expression regulation that involves controlling the process of transcription by controlling the amount of RNA produced by a cell. TATA box, TATA box. Short nucleotide sequence, capital TATA, usually located approximately 20 to 30 base pairs upstream in the 5' direction of the start site of many eukaryotic genes. Part of promoter sequence bound by transcription factors used to stimulate RNA polymerase. CAT box, CAAT box. Short nucleotide sequence, capital CAAT, usually located approximately 80 to 90 base pairs upstream in the 5' direction of the start site of many eukaryotic genes. Part of promoter sequence bound by transcription factors used to stimulate RNA polymerase. Trying to find my spot here. Okay. Next is operons. Gene units common in bacteria, typically consisting of a series of genes located on adjacent regions of chromosome and regulated in a coordinated fashion. Many operons are involved in bacterial cell metabolism of nutrients such as sugars. Refer to the lac operon as a well-characterized bacterial operon. Okay. Next is in the lac operon section, B-galactosidase is an enzyme produced by the lac-Z gene of the lactose lac operon in bacteria. B-galactosidase degrades the disaccharide lactose. Lac-Y, encoding the enzyme permease. Permease is an enzyme produced by the lac-Y gene of the lactose lac operon in bacteria. Permease transports the disaccharide lactose into bacterial cells. And lac-A, encoding the enzyme acetylase. Acetylase is an enzyme produced by the lac-A gene of the lactose or lac operon in bacteria. Next is motifs. Motifs, these regions called DNA binding domains, have folded structural arrangements of amino acids called motifs that interact directly with DNA. Operator. Region of DNA such as that located in an operon that binds to a specific repressor protein to control expression of a gene or group of genes such as an operon. Short interfering RNA or siRNAs. Small 21 or 22 nucleotide double-stranded pieces of non-protein coding RNA, so named because they were shown to bind to mRNA and subsequently block or interfere with translation of bound mRNAs. RNA interference. RNAi. RNA-based mechanisms of gene silencing. These siRNAs are bound by a protein RNA complex called the RNA-inducing silencing complex, or known as RISC, R-I-S-C. Now in the mutation section, dicer. Dicer is an enzyme that cuts microRNA transcripts into short single-stranded pieces of 21 to 22 nucleotides. Next, we define mutagens. Mutagens are physical or chemical agents that cause mutations. Point mutations. A single base change in DNA sequence. Again, that's a point mutation. Next is single nucleotide polymorphisms, or SNPs, SNPs. SNPs, single nucleotide polymorphisms, are single nucleotide variations in the gene sequence, or a type of DNA mutation, the basis of genetic variation among humans. Next is silent mutation. Silent mutation is a base pair substitution that has no effect on the amino acid sequence of a protein. Missense mutation. Missense mutation is a mutation changing a codon to another codon that codes for a different amino acid. Next is nonsense mutation. Nonsense mutations are mutations that change a codon into a stop codon. These usually produce a shortened, poorly functioning, or non-functional protein. Next is frame-shift mutation. Frame-shift mutations are mutations resulting from the addition or deletion of nucleotides that causes a shift in the genetic code reading frame of a gene. Next is inherited mutations. A change in DNA structure or sequence of a gene passed to offspring through gametes can be cause of birth defects and genetic disease. Acquired mutations. Acquired mutations these occur in the genome of somatic cells and are not passed along to offspring. They can cause abnormalities in cell growth, leading to cancerous tumor formation, metabolic disorders, and other conditions. Next is hemoglobin. Hemoglobin is oxygen-binding and oxygen-transporting protein of red blood cells and vertebrates. Mutation of globin genes that encode hemoglobin results in a number of different human blood disorders with a genetic basis, including sickle cell disease. Next, we define epigenome. Epigenome is defined as modifications in an organism's genome that are not due to mutations in DNA sequence. And that completes all the definitions of the vocabulary from Chapter 2 in the Intro to Biotechnology.